Hello students, now we will start with new topic, that is DNA replication. DNA replication, it means DNA synthesizes other DNA, that is new DNA. Now, this total process of DNA synthesizing new DNA is also called as autocatalysis. Autocatalysis. Then what is name for DNA synthesizing other type of genetic material, that is RNA? DNA synthesizing mRNA or simply RNA. Here the process is called transcription. Process is transcription, but the total synthesizing of RNA from DNA is called as heterocatalysis. Means transcription also called as heterocatalysis. Next, DNA replication also called as autocatalysis. There are many processes of making DNA new copy from the existed old DNA strand. 3 ways of autocatalysis are: one is semi-discontinuous method in which Okazaki fragments are formed. Second one, bidirectional method in which both the strands are involving, and this is explained by Y-fork method of DNA replication. And third one is semi-conservative method of DNA replication. Here we take a hybrid DNA strand and we learn the process of how half of the DNA strand is preserved throughout the DNA replication methods. So now we will read these two combinedly with the help of Y-fork. Before going into DNA replication process, there are some enzymes that are required during DNA replication. So first we will see about enzymes involved in DNA replication. We know that DNA is double-stranded helical structure. It is always coiled like this. Now, first of all, these two are connected by the hydrogen bonds. Nitrogen base pairs are always connected by hydrogen bonds. First, the hydrogen bonds should be broken and a gap should be created between the two strands at one point to initiate the DNA replication. So, the two coiled DNA strands should be unwinded like this. So, first enzyme is helicase. Helicase enzyme is used to unwind the coiled strands. Unwind the coiled DNA strand. How it is uncoiling? By breaking the hydrogen bonds. Where are these hydrogen bonds located? These are located between the nitrogen base pairs. We know that adenine is paired with thymine with help of 2 hydrogen bonds. Guanine is paired with cytosine with help of 3 hydrogen bonds. So those are located here like this. If this is adenine, thymine, guanine, cytosine, cytosine, guanine, thymine, adenine. These hydrogen bonds has to be broken down to unwind the strands. Next enzyme is topoisomerase. Topoisomerase, it helps in removal or releasing the tension between the coiling strands. For suppose, this is the DNA strand that is unwinded with the help of helicase enzyme. So think that helicase is present here. Now these two strands are unwinded. Then here on the other side, again the strands, they tend to recoil from other side. That recoiling nature is stopped by topoisomerase to maintain the gap or the distance between these two strands which are involved in DNA replication. This is also called as topoisomerase, also called as DNA gyrase. Topoisomerase releases or removes the tension between coiling strands and maintains the gap between the strands. Next enzyme is third one, DNA polymerase. It is of three types, that is DNA polymerase 1, 2 and 3. So first one, DNA polymerase 1. DNA polymerase 1 is considered to be very important type of enzyme for DNA replication and initiation also. Initiation is done by unwinding helices, but it is also helping in addition of new nucleotides. DNA polymerase 1 mainly functions as a repair enzyme, so called as DNA repair enzyme also. What it will do, there is a small RNA fragment that is helping in addition of nucleotides for the formation of new DNA strands. Now, after formation of new DNA strand, that RNA primer has to be removed or RNA fragment has to be removed. That function is done by DNA polymerase 1. It also helps in removal of RNA primer after the DNA replication. Sorry, you will write here in an easy word, after the formation of new DNA strand. And fills that gap. DNA polymerase, it was first discovered by person Korenberg. So this type of DNA polymerase is also called as Korenberg's enzyme. This also functions as an proofreader. So it is considered as DNA polymerase 1 also does the exonuclease activity. First discovered by Korenberg, so called as Korenberg's enzyme. DNA polymerase also involves in proofreading of newly formed DNA. DNA polymerase also functions as an exonuclease in its 5' to 3' direction and also from 3' to 5' direction. And also from 3' to 5' direction. Next. This is third one. This is fourth. DNA Polymerase 2. This DNA Polymerase 2 is having least priority because mostly when DNA Polymerase 1 and DNA Polymerase 3 are absent, then DNA Polymerase 2 will does both the functions. In absence of DNA Polymerase 1 and 3, this enzyme will work as both. So considered as least reactive enzyme in DNA replication. Next one is DNA polymerase 3, which is very important and main enzyme out of the 3 types of DNA polymerases. It is the main enzyme for autocatalysis, it is discovered by Delucia and Kyrens. It helps in formation of the long DNA new strands. It helps in formation of long new DNA. This enzyme is also called as replicase. Also called as replicase. This DNA polymerase has 3 subunits or 3 domains in its structure. What are those? First one, alpha subunit. Alpha subunit helps in catalytic function, means addition of nucleotides to the newly forming DNA. So, catalytic function. Second subunit is epsilon subunit. It helps in proofreading. Already we have seen DNA polymerase 1 also is involved in proofreading of the new DNA strand. So this is another type of enzyme, proofreading. Next, there are 2 more subunits called as beta subunits. They help in hanging, means the 2 DNA strands are formed complementary, because in case of DNA replication, the 2 strands are behaving as template strands. So, when new DNA is formed, this helps in the hanging of the new DNA strand to the older one. So, this helps as clamps and prevent new DNA from sliding off. So this is about DNA polymerase 1, 2 and 3. We can frame out one more sentence that this DNA polymerase helps in addition of nucleotides to the 3' end of RNA primer for both old strands of DNA. Primer for both strands we'll write. Because RNA primer is attached on 3' end of the old strand. That I will show you diagrammatically now. If this is the new strand that is formed, this one and this one are considered as old DNA strands, and one strand will be continuously formed and another strand will be formed in pieces. So here will be the area where helicase will be binded. Now, here must be the RNA primer. RNA primer attaches new DNA strands on its 3' end. It means the opposite end has to be 5'. then the complementary strand should be having 3' direction. It means that the RNA primer is attached on the 3' end of the old DNA strand. It will be 5', then it will be 3'. Now, in case of other strand, here will be the RNA primer attached. There will be many RNA primers attached on the other side, which is giving discontinuous DNA strands. Then this will be 5'. So, new nucleotides are added on the RNA prime only on the 3' end of the RNA primer. Here we should learn one more point that, for suppose if this is the double-stranded DNA, one strand is called as coding strand. When we are reading about transcription, we will learn this. Another strand is called as template strand. We read that the same copy of coding strand is coded on the new mRNA. Means mRNA strand is complementary to that of template strand. Means here this is helping in formation of new RNA. That process is called as catalysis. Means here one strand is giving its copy, another strand is helping in formation of new strand. Here in case of DNA replication, both the strands are helping in formation of new DNA strands. So both the strands are called as template strands. Means 2 number of template strands. During DNA replication, in case of transcription, we call only one old DNA strand as template, but in case of DNA replication or autocatalysis, 2 strands are named as template strands. All the DNA polymerases commonly does a function, that is, they function in behaving as a polymerization of polymerization activity from 5' to 3' direction, and they exhibit exonuclease activity from 3' to 5' direction. Has polymerization activity of new strand from 5' to 3' direction, 5' to 3' direction, means this direction is of the new strand. See, 5' to 3', the DNA new strand is formed. We can call this one as new DNA strand, polymerization. Next, end. Exonuclease activity in 3' to 5' direction of new strand. It means that a DNA is forming and giving rise to continuous strand here. But if any mistake is there, the reading has to be done in the opposite direction. Whenever new nucleotide has been attached, DNA polymerase 1 has to get involved here and read whether the attached nucleotide is correct one or not. Reading is done from 3' to 5'. Addition or polymerization of the strand will be from 5' to 3' direction of the new strand. Next is single strand stabilizing enzyme. Actually, this is working in the form of a protein. Means this enzyme is having more proteinaceous nature. We know that all enzymes are made up of proteins. This enzyme or the protein part helps in stabilization of or stabilizing the unwinded strands. Then what is difference between topoisomerase and single strand stabilizing enzyme? This is first of all unwinded by helicase. The distance is maintained by topoisomerase. Now, if this strand is giving a continuous new DNA and other strand is giving discontinuous new DNA, then the new DNA should have complementarity with that of old one. So this is old DNA strand and this is new DNA strand. And here only the hydrogen bonds has to be formed. If it is adenine thymine, 2 hydrogen bonds should be in that specific area. The location should not be changed. So that fixation is done by single strand stabilizing enzyme. So we can also write like, I have written here, helps in stabilizing the unwinded DNA strands. It is, main function is, stability means fixedness of the complementary strands. When we call complementary strands, we'll see one example here. So, if this one DNA strand is having adenine, guanine, cytosine, thymine, adenine, guanine like this, the newly formed DNA strand should have thymine, cytosine, guanine, adenine, thymine, cytosine. And this should not be near to that of guanine, because guanine has to form 3 hydrogen bonds with cytosine, and adenine has to form 2 hydrogen bonds. Again here 3, 2, 2, 3. That fixedness or the structural maintenance should be done, and that is because of the stabilizing enzymes that are fixed here. They will not allow the DNA strand to slide off or to move or to change its place or its location. It should be here only. Stabilizing enzymes will be binded here like this. They will help to maintain the fixedness or the correct position of the new and old DNA strands to frame up the correct bonds and to maintain the complementarity. This is about single strand stabilizing enzyme. Next one is about ligase. Ligase is also termed as joining enzyme. In the sense, first we will see about here, an RNA primer will give out new DNA strand, a small fragment, another RNA primer again. Okay. When these RNA primers have given so and so length of DNA strand, later those will be removed by DNA polymerase 1. Then a gap is being created. We are telling it helps in filling that gap. So DNA polymerase allows the ligase enzyme to come and fit here, so that all the pieces of the Okazaki fragments, what we call, or the pieces of DNA will be attached one with each other with the help of ligase enzyme. And that support of the ligase enzyme is given by DNA polymerase 1. Next, it helps in binding or joining of DNA fragments. Next is RNA primer. For DNA synthesis, there is a need of special type of RNA fragment called as RNA primer. RNA primer is a special RNA fragment that helps in binding of new nucleotides for the synthesis of DNA strand. So that helps in attachment of or binding of new nucleotides for DNA synthesis. It means that those nucleotides will be having the composition of pentose sugar, that is deoxyribose sugar, plus phosphate plus nitrogen bases. But this is an RNA fragment. Basically, we know that the nucleotides of RNA fragment will be having the composition of nitrogen bases like adenine, guanine, cytosine and uracil. But the nitrogen bases of DNA will be having adenine, guanine, cytosine and thymine. And the pentose sugar in case of DNA will be deoxyribose pentose sugar. In case of RNA, it will be ribose pentose sugar. But this RNA fragment helps in attachment of the nucleotides for new DNA synthesis. This RNA primer it's a small RNA fragment So it will be having approximately 4 to 15 number of nucleotides. It's made of 4 to 15 number of nucleotides. Means here the nucleotides will be having adenine, guanine, cytosine, uracil, nitrogen bases, and pentose sugar will be ribosome. But this helps in for addition of new nucleotides for the DNA. RNA primer is synthesized by DNA dependent RNA polymerase, or it is also called as DNA primase. Means special type of RNA polymerase supports in synthesis of RNA primer. RNA primer attaches new nucleotides for DNA synthesis on its 3' end. Suppose if this is an RNA primer, its ends are 5' and 3' directions. It will add the new nucleotides on its 3' end. Means DNA synthesis is happening from 5' to 3' of RNA primer and 3' to 5' of old DNA strand. Next is phosphorylase. Phosphorylase helps in addition of a phosphate to the acceptor from an inorganic phosphate molecule. Inorganic phosphate means salts of the phosphates. It helps in addition of phosphates from an inorganic phosphates to the acceptor. Because in case of DNA, we see phosphates are responsible for the formation of nucleotide. If a nucleoside has to get converted into nucleotide, then we need addition of phosphates. These phosphates are binded with that of nucleoside with help of ester bonds between oxygen and 5th carbon position of the nucleus of the pentose sugar. Another enzyme is nuclease. We have seen DNA polymerase 1 is helping in proofreading of the newly formed DNA strand, and also epsilon subunit of DNA polymerase 3 also helping in proofreading of the newly formed DNA strand. Now, third type of enzyme is nuclease. It also helps in proofreading. Of new DNA strand. But this one, for its proofreading function, it needs ATP for energy purpose and magnesium ions. Magnesium ion is working as an activator of nuclease. This ATP will be broken down into AMP and the phosphate molecules are separated so that energy is also released. Converted into AMP by phosphatases and releases energy that is required for nuclease. We will just have a glance regarding the structure of ATP because we have written here some energy is released. ATP full form is adenosine triphosphate. It is also considered as a nucleotide. Nucleotide in the sense it will be having one pentose sugar, one nitrogen base, that is adenine. So carbon 1, 2, 3, 4 and 5. Here it should have 3 phosphate molecules. Here there is less energy in the form of kilocalories. Here there is a little bit more energy. And here also little bit more energy. means more energy. Approximately it will be 2.5 kilocalories of energy at the first phosphate bond. This will not be broken because always this type of nucleotide helps the cell to attach to other phosphate molecules and retain the energy. So this is having some 6.8 etc. kilocalories approximate value. This is not the exact one. The third one, when the energy is increased, then the ester bonds will show some of the bending. Here the pure bending means the bending of the bond between the phosphate molecules will be showing as the bend increases, it means that more amount of energy is present there. Some 8.62 kilocalories of energy at this between second phosphate and third phosphate. So when this bond is broken between the first phosphate and the second phosphate, then this energy is utilized for the enzymes or the substrates to do their function. And also the energy is helping for the formation of phosphodiester bonds between two nucleotides. Energy is also used to form phosphodiester bonds between nucleotides. So these are few types of enzymes that are involved in DNA replication or autocatalysis function. Now we will see the process of DNA replication. Process of autocatalysis. Here the major structures are nucleotides that are framing out new DNA strands. So the cell or the nucleus will check whether sufficient nucleotides are present or not. So where are these nucleotides located initially? Nucleotides for new DNA are located in the nucleoplasm of the nucleus. Now, these nucleotides are of different forms. Not all the nucleotides are involved readily because sometimes they are in inactive form also. Are of active form and inactive form. So we can take some examples for inactive form as AMP means adenosine monophosphate, guanosine monophosphate, thymosin monophosphate, cytidine monophosphate. These are not having sufficient energy levels or the complete structures to involve in the process. So they are converted means activated into by the addition of phosphates. So they are converted into active forms, that is ATP, GTP, TTP and CTP. Adenosine triphosphate, guanosine triphosphate, thymidine triphosphate, cytidine triphosphate. So these are considered as the active forms. The process of addition of phosphates is called as phosphorylation. So we'll write in presence of enzyme, phosphorylase. Next, the site of autocatalysis initiates or site of autocatalysis initiation is called ORI site, origin of replication. The full form is of ORI site is origin of replication. Just we are giving small term like capital O, small r and small I alphabets. Next, what about the number of ORI sites in prokaryotes and in eukaryotes? So prokaryotes have one ORI site. Eukaryotes have many ORI sites. Next. So what is this ORI site biochemically? It is a special nucleotide sequence. It's not a protein or something. ORI site is a special nucleotide sequence. It is not an enzyme or a protein or carbohydrate or something. Small part area on the existing DNA strand where new DNA has to get formed. That is nothing but a combination of special nucleotide sequence, or we can simply say gene sequence, where replication starts. So let's take a double-stranded DNA structure. Initially we know it is nothing but coiled one, and we are reading about the right-handed helix, that is bDNA. We are telling it is existed like this. But suppose take this small part and we will start reading this DNA fragment. Now some part of the DNA is coiled. Now it has to be separated or unwinded with help of helicase enzyme. So this will be helicase. This one, according to NCERT, the diagram is also given by simplifying as, or the name given as Y fork also. This is Y fork. Inverted Y fork, you can write. Inverted Y. This structure or this structure is same. But here we have just written as separated structures. Here will be the topoisomerase. Now, here will be the stabilizing, single-stranded stabilizing enzymes. This is nothing but a small protein part. Now, where is RNA primer located? For the location of RNA primer, we should understand that RNA primer always binds on to the 3' end of the old strand. So if we give this part as 3' and 5', so this will be 5' and 3'. In the same way here also 3', 5', 5', 3' direction. So if we write like this end is having RNA primer like this, then RNA primer will be having 5' end here and 3' end here. We already have discussed that new nucleotides are binded on the 3' end of the RNA primer. So this strand gives continuous new DNA strand. When there is continuous new DNA strand, this is called as leading strand, which gives continuous new DNA strand. But here, this is the 3' end on the other template strand. So RNA primer has to be here. It is giving out new nucleotides which are attached. Okay. So it is giving discontinuous strands. This is how we will discuss later while drawing the structures. So here, this is giving discontinuous strands of the new DNA. So this is called as lagging strand that has discontinuous new DNA fragments. So this is the basic information regarding the Y fork. Now we will see how the DNA autocatalysis is getting extended and how the new DNA strand is continuously formed. Think that initially the inverted Y fork is like this. So this is 3', 5'. Again this is 3' and 5'. We will decrease the length of this one to show the discontinuous strands. RNA primer will be attached here. So RNA primer 3' end is towards the coiled fork. So new nucleotides are attached here. On the other strand, RNA primer is giving some sequence like this. So this area is called as ORI site. ORI site. And this is RNA primer. In the next stage of extending of new DNA strands, we know that helicase has to unwind. so that this will keep on unwinding like this towards the upper direction. So the length of this one will be increased. The unwinded part will be increased. Think you can find out the difference between the length of this and also the length of this is more. So already we have written here 3 prime, 5 prime, again 3 prime and 5 prime direction. RNA polymerase, initially we have written here only 2 number of nucleotides. Now we are increasing the number of nucleotides that are added. But how many nucleotides are present here? RNA primer, which is on the 3 prime, only 3 number of nucleotides. Then again the strands were unwinded, means again the new RNA primer will come and bind here. It also will give addition of some new nucleotides there, because this RNA primer will bind new nucleotides only on its 3 prime end. On its 5 prime end it will not bind. So new RNA primer will come and fit on the other strand, which we are calling as lagging strand. This is leading strand. This is ORS site. So here 3 number of nucleotides have formed into one DNA fragment. Other RNA primer, other nucleotide sequence. Now here is a gap. Again 2 number of nucleotides can be added on the 3 prime end of the RNA primer. Again, if the unwinding kept on increasing because of the helicase enzyme, then the length will be much increased like this. So this is 3 prime, this is 5 prime, 3 prime and 5 prime. RNA primer is here, which is having 5 prime to 3 prime. In case of leading strand, new nucleotides kept on increasing without any disturbance on the 3 prime end of RNA primer towards the 5 prime direction. But here 3 number of RNA primers are there. So another RNA primer, another RNA primer, another RNA primer. In this way, the length will be increased for the continuous strand in a continuous way. But on the other side, RNA primers are binding new nucleotides on their 3 prime end. But since the unwinding is not allowing to add nucleotides on the 5 prime end of the RNA primer, it has to arrange new RNA primer there, and a new DNA fragment has to be formed. So here we have represented some 4 number of DNA fragments. All these discontinuous DNA fragments are called as Okazaki fragments. Okazaki fragments. Now this is continuous DNA strand. Which is supported by the 3 prime end of the RNA primer. Now, after formation of certain length of the DNA, this will get separated as a new DNA strand. But what about this one? The discontinuously formed DNA fragments. The RNA primer is removed with the help of DNA Polymerase One, and it tries to fill the gap by attracting the ligase enzyme there. Ligase enzyme is, its function is nothing but joining of different nucleotides and framing out into one complete strand. So here we can frame out DNA Polymerase One will break RNA primer from nucleotides and allows ligase to join the nucleotides. Means here the DNA polymerase is functioning here also. DNA Polymerase One is involved. Now this strand and this strand both are functioning as template strands. Now, if a question has come, how many number of phosphodiester bonds are there in a discontinuous DNA strand with 4 DNA fragments like this? Or in an Okazaki fragment of, we'll write one question here. In an Okazaki fragment of 5 nucleotides, how many phosphodiester bonds are present? If such type of question was asked, then how to answer? For suppose think that this end is having 5 number of nucleotides. 2 nucleotides side by side are attached by phosphodiester bonds. So how many will be there? On one side 4, other side 4. Just decrease the number to 1. So if this is one nucleotide, other nucleotide, other nucleotide, other nucleotide. If a nucleotide has to be attached on its one side, 5' end, we know that 2 DNA strands are oppositely running with each other. So this is one nucleotide strand, and here will be other. But here Okazaki fragment is having only one single strand. So 1, 2, 3, 4. So 4 number of phosphodiester bonds. If the number of ester bonds are asked, it will be 8 in number. 8 number of ester bonds. Next. Okay. So this will be the one model question regarding the number of Okazaki fragments and number of phosphodiester bonds. Clearly we have to think about whether the question is about a single strand or about the 2 double strand. If a term was given between complementary strands, then we should double the number of phosphodiester, because we know that in case of DNA, the 2 strands are running anti-parallel to each other. Complementary means one template strand has to be there, one new DNA strand has to be there, or one template strand and one coding strand has to be present. Then we should consider the question is about double-stranded helical DNA. But here the question asked is about Okazaki fragment, means only one single strand. This is about DNA replication.